A critical question is, how do we determine which genes are on or off? We wouldn't be able to have a difference between, say, a nerve cell and a muscle cell if we didn't do that. An embryo wouldn't be able to decide, put the nervous system here or put an arm here. And as we compare the differences between, say, a normal cell and a cancer cell, clearly many diseases such as cancer involve not regulating genes correctly, turning genes off when they should be on, or not turning genes on when they should be. So how do we regulate genes? The answer is lots of ways. Some of the most important ways are in the regulation of transcription, and that's what the first part of this 2-video sequence covered, the regulation of transcription through making DNA heterochromatin versus euchromatin.
There are several steps which must occur before.
Or producing the transcription factors needed for transcription, or by adding methyl groups. So that was how transcription was regulated. This video is going to talk about the later steps on how to regulate gene activity, the post-transcriptional regulation, the translational regulation, and the post-translational regulation. One of the ways that post-transcriptional regulation can work, there are several, is that if you recall, once transcription occurs, a couple of steps have to then follow. One, a GTP cap is added. A series of adenines are added. That becomes important in the next step, what's called the poly-A tail. But then finally, there are intervening sequences known as introns, which are removed. And so just because all of these were copied from the DNA message doesn't mean that they will all become protein, or the code for a protein. So this section will be removed, another section will be removed. Those are what are called the introns, and thus the remaining sequences, the exons, are combined, and that then produces the mRNA, which leaves the nucleus and is translated. But when those introns are removed, it turns out that this provides an opportunity to regulate the gene, all right, because the introns, their removal can vary. And so one single gene could actually then encode multiple different proteins. They would be similar, but they would be different. So for example, take this case here. If the introns are removed, let's say that here's the first exon, and if it is translated, it produces a sequence of amino acids that looks like this. Here is the second exon. It would produce a series of amino acids that looks like this. Here's the third, etc. And so we could make a protein that has all of these sections, but we don't have to. Just because these are all possible exons, a specific cell could just use the first three, and then its protein that it translates from the gene in question looks like this, with the pink, the green, and the aqua portions. However, another cell in the body could take the same gene, but then splice together different exons, so that instead of just having those three parts, all right, which might exist in, say, one cell type, you might get all five. And so when you ask, do, say, liver cells, do white blood cells, do brain cells use this gene? The answer could be yes. All of those cell types use this gene. But what does the protein look like in each of those cell types? It could vary from cell type to cell type, because one cell type could splice the introns, splice out the introns in a different order. And so we can have this alternate splicing, so that one gene can actually produce different amino acid sequences based on which of the sections are spliced together. That allows different cells in the body to specialize a protein for their functions. And also, it might also allow for the creation of new genes in what's called exon shuffling, so that, the exon 1 of gene 1, the exon 5 of gene 2, the exon 3 of gene 3 might actually end up at some point being combined to make a new protein product. There are examples known of that, like LDL receptors. And so this is one way of regulating a gene because the the splicing of the introns can then be varied and produce different topics. So once again, if these are all different exons, we could take different combinations of these. And even though it's one single gene, this could then give different protein products. Now, most human genes involve introns. Introns can represent more than half of the length of a gene. And at first that seems very wasteful. Why would we go to the purpose of transcribing all of these nucleotides if we're just going to throw them away if they never become protein? Well, it seems that this alternate splicing is actually very important. And when we think back in the evolution of the eukaryotes, it seems like introns which originated in the endosymbionts, the organisms which would later become mitochondria, it seems as if their introns then gave rise to the spliceosomal intron. So it seems that this was an invention of the early eukaryotes. And since the typical eukaryotic gene then has these introns which can now be spliced together in different ways, this seems to be, a critical point in the evolution of eukaryotes, and certainly the higher eukaryotes. All right, so that happens after transcription is done, and that's one way that we can continue to modify genes. There are other ways. So let's say that a transcript makes a series of nucleotides. Well, the nucleotides can actually be modified, right? So they can be changed, as I'll get to, but there are more nucleotides than just adenine, guanine, cytosine, and uracil, since we're talking about RNA. You can make all of these variants. These variants are one of the reasons that the tRNAs have such unusual shapes, because these variants, they don't pair the way that, say, adenine and uracil will, or guanine and cytosine would. And so making new RNA nucleotides can certainly change. And that is a subclass of this is what's known as RNA editing, so that you can take a specific nucleotide and the DNA said, put this nucleotide here, but you can then subsequently change it in a way that the DNA did not code for, so that if this becomes protein, the protein amino acid that would be made is not what the DNA called for, because you edited the RNA after it was transcribed. So this is a form of post-transcriptional regulation. One of the important forms of RNA editing involves the creation of inosine, which is a modified nucleotide if you inserted a nucleotide where one had been lost, that could now actually restore normal function after a frameshift mutation. But if you convert adenine into this inosine, this can change base pairing, all right? And so that could be significant. But then this could also change how the RNA molecule folds upon it itself. This can be a defense against viruses, because we can then modify the nucleotides of double-stranded RNAs, which are ones that viruses use, but there are many places in the human genome where we edit RNA turning adenine into inosine. And there are enzymes which do this, and the dysregulation of this can cause a number of disorders, including cancer. So we can edit nucleotides. And this is significant because it can change amino acid order. It can change the splicing of introns because we can change the structure of the intron. And it can change how RNA molecules fold upon each other. So that is significant. Another point, and so I'm going to make one point and then go into a related one. When we think of our DNA, we have a lot of it. And if we think of each of these red pieces, there's 100 of them, as representing 1% of what's called your genome, then, so this is the DNA. If you were to ask what percentage of this is transcribed into RNA, well, not all of it, but most of it. So, I mean, this could vary, and it certainly varies from cell type to cell type. But let's say 70% of this is then the transcriptome. So we've made a lot of RNA, but only a small subset of this will actually become code for protein. Only about 3% of it codes for the proteome. So just because RNA is made, it does not have to become protein. And actually, when you think of the genome, most of the transcribed RNAs of the transcriptome do not become protein coding members of the proteome. All right, so that's one point. And then a related point is transcription occurs in the nucleus. But so the transcription occurs here, but translation is occurring at the RER here in the cytoplasm. The only way that translation can occur is if the transcript leaves through a nuclear pore to go into the cytoplasm. But we control that. And it seems about half of all transcripts don't leave the nucleus, all right? And that they could be sitting there waiting for the right signal and say, the right stage of the cell cycle or some things could then be the signal for an mRNA transcript to leave the nucleus and then to be translated. So transcription can occur, but this subsequent part of post-transcriptional regulation needs to occur before that can be converted into protein. You have to decide whether or not it leaves the nucleus. And just because an mRNA has left the nucleus doesn't mean that it will be translated into protein. One of the reasons is you could destroy it, all right? Because there are these small RNAs known as microRNAs. This would be part of a bigger topic, as would many of the topics here. I'm just going to quickly, give an overview here. The microRNA could then target an RNA. And once a microRNA binds to it, this would then target that mRNA for destruction, right? And so even though a transcript has been made and there is the possibility of the translation of this mRNA, we could then have the microRNA bind a complementary sequence on an mRNA, and this would then target it for destruction in what's known as an RNA-induced silencing complex. This can cause the degradation of the mRNA. This could prevent it from being translated. So just because the mRNA was made doesn't mean that it will actually ever be used. And we have specific microRNAs, which will bind to specific genes. So for example, let's say you make the mRNA for P53, which is a tumor suppressor, and you're thinking, oh, this is great, I'll decrease the risk of cancer by making P53. Well, microRNA 125B can bind to this and inactivate it. So just because you've made P53 doesn't mean that it will become protein and be used. You can actually eliminate it. And there are many other examples known as well. So when we talk about epigenetics later, the degree of methylation which we employ can be varied, because here is an enzyme or the transcript for an enzyme which for a DNA methyltransferase. But this microRNA can then bind to it and target it for inactivation or destruction. So once again, the transcript was made, but we are still regulating it. So there are different levels of regulating genes, and what I've just mentioned are some of the ways that can occur after transcription, but before translation, we can still regulate how active a gene will be. The next step is that translation can be regulated through a couple of means. One is, as I mentioned before, the transcript has adenines added to the end that the DNA did not code for, what's known as the poly A tail. Poly meaning many, so look at all of the adenines here in a row. They were added afterwards, and once again, the DNA did not code for that. Well, how many adenines are made? Well, that varies. We can vary that. And the length of the poly A tail will determine how long this can stay active in the cytoplasm. The longer this lasts in the cytoplasm, the more protein could be translated from it. So a short-lived mRNA would make a little protein, a long-lived mRNA would make more. And we have a say in regulating how much protein would be made from an mRNA by the length of the poly A tail, which is made from it. There are other ways in which translation can be regulated. So for example, it can be paused, it can be controlled. So here's one example. Some proteins are translated by ribosomes floating in the cytoplasm, while others might begin their translation in the cytosol floating around. But when a certain sequence is made in the amino acids, what's known as a signal sequence, okay, this could then bind to a protein. All right, so here we have the signal recognition particle, the SRP, which binds to that signal sequence. And it will now pause translation. So here you have the ribosome, it's translating the mRNA, making a protein. But now after the SRP binds the signal sequence, that will pause. And now the complex will be transported to the RER, the rough endoplasmic reticulum, and now translation will occur there. And so here we are, we're regulating translation to a degree in that we're pausing it so that this can be migrated and translation can occur at a specific region of the cell, maybe because the protein has to be folded in such a way, and that can only occur in the lumen of the RER, where the pH is different. And so now, because it was transported here, the rest of the amino acid sequence will be made there. Okay. There are other ways in which translation can be regulated. One of the fairly new changes in our understanding of the ribosome is that all of those little proteins in the ribosome, the ribosomal proteins, they can vary. So while, it's the rRNA, which is actually performing the enzymatic reaction where those amino acids are joined, we know that there's more to a ribosome than the rRNA. So the small subunit can have 21 little proteins, the large subunit 34 proteins. This is true of prokaryotes. In eukaryotes, it's even more. So there's all of these little proteins. What do these proteins do? Well, some of them are standard and they are always present. So apparently they, just are essential for the function of ribosomes. But then other ribosomal proteins can vary. All right. So some ribosomes have these proteins, other ones don't. So ribosomes then come in different classes that may then preferentially translate different mRNAs. So certain mRNAs may be translated by ribosomes with some of these small ribosomal proteins, and others are translated by other ones. And so, for example, the HOX proteins expressed in fetal development, there are ribosomal proteins which will preferentially translate HOX proteins or proteins for white blood cell development. So here you can see just the names of different ribosomal proteins, they're numbered 38, 21, 5. They can make part of ribosomes, but not every ribosome, just the ribosomes which are translating HOX genes, or just those making body hair or involved in the differentiation of white blood cells, etc. So translation can then vary by the proteins which are actually inside the ribosome. And then in addition to that, there can be what are called RAPs, ribosome associated proteins. All right. So there can be other proteins which are associating with the ribosome, which will have some function in translation, maybe encouraging it, maybe suppressing it, right? Maybe pausing it. And so the other proteins can then change translation in one way or another. Once again, these ribosome associated proteins. Some of these can interact with that poly A tail, and degrade mRNAs after a certain amount of time. Some of these proteins might only be made at certain stages of the cell cycle. And so a ribosome needs to be fully functional, a ribosome associated protein that's only made in G1. So when the cell decides, hey, it's time to divide, we're going to progress through G1, then the ribosome associated protein is made, which then causes the translation of the mRNAs for proteins active at that time. There is a specific ribosome-associated protein, which is only known in metazoan animals, which may have a role in their complexity as well. And then, we could find other examples. So, for example, if you ask, how does an amoeba move? All right, I mean, how does it kind of like shoot out these little arms? Well, the answer is it's going to, dissolve its cytoskeleton on one side of its body and then assemble a new cytoskeleton in the pseudopods and then flow into it. Well, how does it do that? Well, the actin mRNA is transported to the new area where it's going to grow. So if it's kind of flowing into this one area, the mRNAs are transported there, and then translation occurs there. All right, so this amoeba is building a new cytoskeleton in front of itself because mRNAs are translated there and translation occurs there. So here you can see translation is being is being regulated. Translation can also be paused. So for example, in eggs which have not yet been fertilized, all of the transcripts could be there, but translation could be paused until fertilization, and then translation is allowed to proceed. So just because an mRNA leaves the nucleus does not mean that it is automatically then going to be translated. Translation can be paused, it can be suppressed, it can preferentially affect certain mRNAs, so we can regulate translation. Last point, we can then regulate the proteins even after translation is done. So there is a post-translational level of gene regulation. Just because the protein is made, all right, doesn't mean that we're done regulating it. One of the main things is many proteins get sent to the Golgi because the protein won't work until the Golgi is done changing it. So maybe a couple of amino acids have to be removed. So for example, in the case of insulin, when insulin is made, in translation, it's not functional. It has to be sent to the Golgi and a couple of amino acids removed from one end before it can be functional. Very often in the Golgi, sugar groups have to be added, and without these sugar groups, the protein is not functional. All right. One very important aspect, and this is very important in our understanding of cancer, is your cell is full of these proteins. And if you're asking, are they active at this moment? The answer is no, they might just be sitting there doing nothing. And if you ask why, the answer is because we activate proteins by adding a phosphate to specific amino acids. So we have enzymes called kinases, hundreds of them. And these kinases will then take a phosphate and attach it to specific amino acids, like a serine, like a threonine, like a tyrosine. And when we attach this phosphate, the protein is now on. So proteins can be made and just sit there and do nothing because there's an on-off switch. And it is not until we add the phosphate that then the protein is now on. So we have kinases. There are serine kinases, which add phosphates to serine amino acids. There are receptor tyrosine kinases, very important in the animals, right? A major group of enzymes which gave animals their signaling pathways are the receptor tyrosine kinases, which add phosphates and then can turn the protein on. So a lot of proteins then, once the protein has a phosphate on a specific amino acid, it is now on. And then there are enzymes called phosphatases which remove it. So not only can we add phosphates to turn a protein on, a kinase would do that, but we can remove the phosphate, and that would then turn the protein off, and phosphatase enzymes do that. One of the ways we regulate proteins, once again, is in the Golgi and in the RAR. We can change the structure because, making a protein, if it doesn't have the right shape, it may not be functional. And as we do this, it's being measured and checked, because if it doesn't assume the right structure, we can actually then destroy it. So proteins that don't fold correctly, forming the right, disulfide bonds or salt bridges, et cetera, they can be destroyed because, they are inappropriate. If too many of these accumulate, a cell could actually kill itself or be destroyed by white blood cells because of abnormally shaped proteins. And so we can regulate, proteins in their folding. And then there are so many other ways. I just want to give a couple of examples and then wrap up. So for example, the cells that make collagen actually don't make collagen, they make pro-collagen, right? And then pro-collagen then is assembled outside the cell, all right? And so then that's obviously something that can be regulated. So we make, the pro-collagen precursor, but its final structure is not determined until we then have additional enzymes act on it. And then plus, if you'll notice that collagen is made of a triple alpha helix. Now notice I had different colors there. All right, they don't have to be different colors because you could have one collagen gene code for each of the 3 triple helices, which then get joined. But you could then use 3 different collagen genes. And so a lot of proteins are functional when they clump together in their quaternary structure. So you could have different subunits form. So what does your hemoglobin look like? Well, if you make beta hemoglobin and you fuse it with alpha hemoglobin, that's one thing. But if you still make gamma hemoglobin, now you're making fetal hemoglobin. So you can have different subunits fused together. There's lots of different collagen genes. Which collagen subunits you use can then make the collagen that you find in cartilage versus blood vessels, etc. In the digestive system, lots of enzymes are made in an inactive form, but then get turned on. So for example, pepsinogen is what the stomach produces, but when you put that into the acid of the stomach, it will cut off some of its amino acids and now become the digestive enzyme pepsin. So the cells make pepsinogen in the inactive form, but then in the right environment, in an acidic environment, it then becomes the active form of pepsin. There are other ways. So you can, once again, here are a bunch of digestive enzymes which would act in the small intestine. And if you remove amino acids from them, then they become activated. And so the pancreas makes inactive enzymes that then go to the small intestine, where this sequence is cut off. A valine with 4 aspartic acids, and then there's a lysine. So when you cut that off of, say, trypsinogen, it then becomes the active digestive enzyme trypsin. So once again, here we are regulating a gene. It wasn't done in the form of trypsinogen that was released by the pancreas. It's not until it gets to the small intestine and that section is cleaved off that it becomes trypsin. Okay. So there's obviously so much more, but this was meant to just be an introduction. And so we regulate our genes. It's not just a question of what genes you have in your genome, but which ones you're using, all right? Are you transcribing these genes? Are the ones you transcribed, did they leave the nucleus? Did you translate them? After you translated them, what happened in the Golgi? Did you make, add the right sugars, as hopefully will come up in the next video? And so this is a critical a topic in genetics. How do we regulate our genes? And it turns out we have so many levels of regulation. We can regulate it at the transcriptional level. We can regulate it at a post-transcriptional level. After it's transcribed, we can regulate its translation. And then even once the protein is made, there is still post-translational regulation, which is possible, say at, the Golgi, for example, just in what sugar groups. I just want to mention this because this video was paused. Anyway, so we have all of these ways of regulating genes.